I have the great pleasure of welcoming Todd Coleman to our virtual stage. Todd is a professor in the Department of Bioengineering at UC San Diego. A very exciting development is that Todd will be joining the Stanford faculty next year as a professor in the WuXi Neurosciences Institute and the departments of bioengineering and electrical engineering. So yay for that. And thanks to the WuXi Neurosciences Institute for making that happen. Just a little background on Todd. He studied electrical engineering and computer engineering at the University of Michigan, and then went on to complete the MS and PhD degrees in electrical engineering at MIT. So at this point, Todd was a straight up engineer with deep expertise in control systems and information theory. He then went through a transformational postdoctoral fellowship in neurosciences at the Mass General Hospital and MIT with Emory Brown. Todd then became an assistant professor of electrical engineering and neuroscience at University of Illinois, after which he joined the faculty at UC San Diego. Todd and his collaborators have developed wearable bioelectronics that bend with the skin and can sense a variety of modalities and then transmit that information, a rich set of information, wirelessly for analysis. Recently, Todd's been developing technology for non-invasive monitoring of the digestive system to understand its interplay with the nervous system. And that's going to be the topic of his presentation today. Todd, I'm just really happy you could join us for the symposium today, and thrilled that we'll be welcoming you to the Stanford faculty in the near future. So I'll pass to you.
Thanks so much, Scott, for the introduction. And yeah, I'm happy to dive in. And as Scott alluded to, I started off my career, undergraduate studies through my PhD, unapologetically, just really as an electrical engineer and applied probability person. And very thankful to my graduate advisor, Muriel Medard, who gave me the best piece of advice in my career when she strongly encouraged me to do a postdoctoral study in something wildly different. And so I went on to do a postdoctoral study under Emory Brown, where I was able to use the fact that he has a PhD in statistics to learn basically neurophysiology through the language of statistics. And so I began to operate at the intersection of neuroscience and data analysis. And that process also got involved in technology development. And some of the things that we have sort of been involved in include, from the pure analysis perspective, doing high dimensional statistics and how you can transform samples from one probability distribution to another. We've been building these thin, stretchable electronic sensors, as Scott alluded to. And then we've sort of integrated, operating at the intersection of all three of these areas, just as an example, and building wearable devices that can track sleep with just a patch that you mount on their forehead, simultaneous monitoring with EEG, validating that it's effective, and then building automated algorithms that can actually stage sleep every 30 seconds. So the purpose of my talk today is within the context of the digestive system. And my thought process in moving in this direction began, unfortunately, when I lost my dad, shown in this picture here with me, 2011, to pancreatic cancer. He had lost his mom to stomach cancer. And so although I had absolutely no expertise in the digestive system, after my experience working under Emory Brown, I had, there's a saying I come up with, feeling comfortable, feeling uncomfortable. I felt comfortable actually exploring this new area. So when we began to explore this, what's interesting is if you take a look at someone who has typical symptoms such as nausea or bloating or abdominal pain, you could have a variety of all of these different conditions as shown here. And what's truly remarkable about this is the path towards getting an effective treatment goes through your primary care doctor, followed by a GI specialist, followed by usually having a very long relationship, even many situations over years with your GI specialist, before they can pinpoint what the issue is and come up with a treatment. And so it should not be surprising to us that this comprises, about $150 billion US annual costs. And this is the second most common reason that someone misses school or work after the common cold. So at this point, me as an electrical engineer, done some neurophysiology, I still don't know how I can contribute, especially because when we look at the digestive system, we think about, well, it's clearly mechanical, because the process of peristalsis is squeezing food through. And also there's all sorts of chemistry and things and whatnot that are taking place to break down foods. But we came to realize that there's an enteric nervous system, a nervous system of our gut with over 100 million neurons. And further, which is called the second brain in China, that second brain is connected to our true brain in many ways, some of which are electrical. And so, for example, our spinal cord as well as our vagus nerve provide bidirectional information flow between our digestive system and our brain. So as you can imagine, we began to become sort of more intrigued. And sort of speaking to that link between, for example, our brain and our digestive system, we have our sympathetic nervous system, which we know will increase heart rates, but it turns out it hits the brake pedal on the digestive system, and the parasympathetic nervous system associated with rest and digest, that actually slows down the heart and it hits the gas pedal on the digestive system. So if we start to dive more into this at a high level, we can, if we focus on the stomach for a moment, we have the sort of top part of the stomach is the fundus, the middle part is the body, the bottom part is called the antrum, and then we have our pylorus, which is a sphincter or a gatekeeper that allows things through before they go to the small intestines. And the typical process of when you eat food, it should be that after it goes to the top part of your stomach, food gets to the antrum sort of region, and there are these very large amplitude contractions that occur to pump the food through. And when the food is sufficiently broken down, it gets released to our small intestines. There are some people in one GI condition, for example, is called gastroparesis, which is when the food basically stops in the stomach for a very long period of time. So from a sort of neuromuscular perspective, it turns out that as I mentioned, we have those 100 million neurons, and clearly we have these muscles in our nervous system, but there are these sort of conductors or maestros, as I call them, in between, which are these interstitial cells of Cajal. And so these are cells that have a membrane potential, and when it crosses a threshold, it generates action potentials, and those action potentials are actually connected to our smooth muscle cells via gap junctions. And so what's interesting about these cells is that they always have a slow wave that's oscillating. So slightly different from neurons, they have the membrane potential is slowly oscillating, and that membrane potential, when it crosses the threshold, it generates action potentials, and it can be stimulated, or the membrane potential can be shifted upward by acetylcholine or information coming from the parasympathetic nervous system, and it can be reduced by information coming from the sympathetic nervous system and norepinephrine. So there's this very dynamic process, and there's the interplay with the vagus and the spinal cord, as well as from the enteric neurons. So just to emphasize, these pacemaker cells have this background oscillating slow wave that's always occurring at a very slow frequency of 0.05 hertz. And so what this means is that when it begins to cross a threshold, we know exactly when those action potentials are going to occur. They're going to occur at the local maxima of those. And that is connected to our muscle tone via gap junctions or electrical synapses, which actually create the peristalsis or the contractions. What's also interesting about these is that if we were to watch sort of a simulation of this occur, we have multiple bands, and they're propagating basically from the top of the stomach down, coordinating the sequence of contractions towards the small intestines. So this is a bit of the electrophysiology of what occurs. And if we stand back and take a look, now that I sort of explained this, what could be some of the underlying causes of people with upper GI symptoms? Well, it could be the top part of your fundus is not stretching as it should when food first comes in. It could be that those pacemaker cells that I mentioned are not operating the way that they're supposed to. Maybe they're not coordinating the slow wave from the top down. It could be that the amplitude of that slow wave activity in the antrum, which is where the largest contractions occur, is not sufficiently high. It could be that the gatekeeper, or the pylorus, which separates the stomach from the small intestines, is not opening and closing as it should. It could be that autonomic nervous system information coming down from your brain is not operating accordingly. Obviously, it could be that something in your duodenum, which is the top part of your stomach. If you're very sensitive and you have lots of pain, maybe it's because your viscera just more generally are hypersensitive, or not surprising, and this actually happens quite often, it could be that you have psychosocial stress, or you have some other type of central nervous system abnormalities. So unfortunately, some people sometimes get sent to the psychiatrist if they cannot get their GI symptoms resolved. So as you can start to see, it takes a very long time to pinpoint these conditions. And so the natural idea that came to our mind is if we take a look at what the EEG has done for the brain, what the EKG has done for the heart, could we place some electrodes over the abdomen to do something analogous for the stomach? And so I collaborate with my colleague Dave Kunkel on the Department of Gastroenterology, and we sort of wanted to play around with this idea. And as with many things, you're not the first person to come up with this idea. And so it turns out this idea of doing this got popular, very popular in the 1980s, so popular that actually a billing code even got generated, and so you can bill CMS to this day for this. And the idea that was recommended by the doctors in the 80s was to place one electrode at the midpoint between the xiphoid and the umbilicus, and then place a pair of other electrodes, the ground and the reference, to the left and right. This will generate one waveform. And as I mentioned before, when food first enters the stomach, the top part of our stomach, our antrum, stretches. Those stretch receptors send information to our enteric neurons, and downstream, they provide acetylcholine to our pacemaker cells in our antrum to get ready to contract. And so what that means is that 0.05 hertz oscillation should be increasing in magnitude after you have a meal. And so what if we just did a time frequency analysis? Do we see more energy post-meal? Makes a lot of sense. Unfortunately, a lot of papers came out after this got popular in the 80s showing that this does not correlate with invasive clinical gold standards and has doubtful clinical utility. As such, people tried to do this but didn't get meaningful information. And so one thing that Scott didn't say about me is that I'm originally from Dallas, Texas. And if you know about the Dallas pro sports teams, there's the Dallas Cowboys for football and the Dallas Mavericks for basketball. And so in the spirit of being a Maverick, just because people tried and they failed, that didn't stop us to explore doing this. Especially what came to our mind is that this stuff got popular in the 1980s. Technology and signal processing has improved tremendously since then. So we wanted to take a fresh look at this. So in partnership with my colleague, Dave Kunkel, one of the first things we did is, so we got some CT scans from some of his patients, and we identified where those three locations of the electrodes would be. And as you can see in this plot where I'm placing my cursor, in the two middle CTs, the three red dots don't cover the stomach. And so there's a saying that noise in equals noise out. And so it turns out there's a lot of anatomical variability from one person to another in terms of the location of their stomach. Something else that occurs is that when you place electrodes on the abdomen, all sorts of small artifacts can take place. And so one of the first things that we did is we said, well, hey, let's use a multi-electrode array to make sure we're covering the stomach. And secondly, because this signal is always at a 0.05 hertz, if it's get masked at certain points in time, we can identify those time points very easily because there's lots of broadband energy, and we can develop a variety of signal processing algorithms to eliminate that. So there's a saying I like to say that the electrogastrogram, the electrophysiology of the stomach is what I call the Rodney Dangerfield of electrophysiology. He gets no respect. And so if we want to convince doctors or clinicians to use this stuff, we are operating below sea level, and we need to first get to sea level. So one of the things that we wanted to do to get to sea level was to do a validation with a clinical gold standard. And so a lot of people, after they go to their primary care doc and their symptoms cannot be resolved, they go to a GI specialist, they do an endoscopy, they don't see anything wrong. They send you over to nuclear medicine and they have you eat a radioactive egg sandwich. And basically that radioactive egg sandwich, they image your abdomen, and that radioactive egg sandwich is going to be emitting photons. And the nice thing about this is that it's either in your stomach, and after it clears your stomach, it goes directly into your urine, into your bladder. So it's either in the stomach or it's not. So this allows us to see how long this egg was in your stomach before it cleared. And so typically it takes about 4 hours. Sort of the idea that we had is that, well, this is different than taking a CT, because every time you take a CT, you're exposing someone to more radiation. Here, the radiation is from the egg sandwich, not from the imaging. So we can image as much as we want. So although you typically image at the 1 hour mark, the 2 hour mark, and the 4 hour mark, we said, let's image every second. And so we got IRB approval to do this. And as we image every second, we might be able to see the mechanical process of the contractions occurring in the antrum. And indeed, we can see that. And so here's sort of a plot, and you can see if we just build a region of interest over the antrum, these 4 dots on the plot, by the way, are fiducial markers from our electrode array. And so this top plot here is basically just the intensity in a certain region of interest over time, and you can see it's oscillating. Then the bottom plot is just the voltage of one of our waveforms associated with the electrogastrogram. And remarkably, in this subject, you can see that there's a dominant peak at 0.05 hertz, or in the vicinity of that, for both the mechanical and the electrical readout. And further, in 6 subjects, they're equivalent up to the 3rd decimal point. So this is just us trying to get to sea level from the Rodney Dangerfield perspective. So when we place the electrodes appropriately, we use an electrode array, we can get more robust information, especially when we eliminate the motion artifacts. So the next limitation that we surpassed compared to what was done in the 1980s is that if you have a single waveform, anyone who knows physics is you cannot be able to understand the spatial properties of the slow wave and peristalsis and whatnot. You might ask yourselves, well, why did we think this was important? Well, luckily, we started playing around with this project in 2011. And in the next year, a big paper came out in the journal Gastroenterology. And in this journal, they basically looked at patients that were undergoing abdominal surgery anyway, and then they placed electrodes right on the surface of the stomach. And there were some patients who had GI problems and some people who did not. In the patients who did not have GI problems, they built vector fields of the direction that the slow wave was propagating. And lo and behold, just as you expect, it's propagating like from the esophagus towards the small intestines. There were other patients who had diagnosed GI conditions, and lo and behold, what they found were interesting things like an abnormal initiation, where the wave is being originated basically not where you would expect it to be, and so you get this bifurcation of propagation. And so this visualization sort of makes this clear. The wave initiation point is not at the top, but rather in the bottom or in the antrum region. And so it's not surprising if someone has nausea or reflux or other issues. So we came up with this idea of, well, let's use our multi-electrode array and let's see if we can actually use the multiple electrodes, not just to make sure we cover the stomach, but also to extract spatial information. So what you see here is just a plot of voltages from 3 different electrodes. And just as we would expect, if there's a wave propagating, we should see phase shifting relative to different electrodes. And if I make a plot of this, you can sort of see this clear. You can see very clearly that there's this wave that's propagating towards the small intestines. And we can do a little bit of math to capture all this stuff statistically using the types of techniques that I learned in my PhD. We can extract the phase, we think statistically. So we want to make sure that this phase properties that we see or this direction cannot be described just by chance. So we use this measure known as the phase gradient directionality, which by the way is also used for ensemble LFP recordings with multiple electrodes. It's used in the neuroscience community to make sure that you see an actual plane wave propagating. And then we ask ourselves, over the time points where we see a statistically significant plane wave propagating, what direction is it moving? Okay, and so for this example, you see most of the time, this circular histogram shows that it's moving towards 180 degrees. So we did a study with my colleagues' patients where we had CT imaging on every patient. And so we placed the center of the electrodes over the antrum. And then we basically ran our statistical analyses and looked at what fraction of the time are the slow waves propagating in any direction that are significant. And interestingly, within the context of a control, you see this very narrow histogram pointing towards 180 degrees. And it turns out that is the direction towards the small intestines. Here we have a patient with gastroparesis, and the direction towards the small intestines is again around 180 degrees. But this histogram shows that we're not seeing any, very seldom are we seeing spatial propagation in that direction. So the field of GI compared to other fields still has a long way to go. And I guess what I mean by that is before this result that I'm showing in front of you, there was not a non-invasive way to build an objective measure of GI function that correlates with the severity of symptoms. And that last point is important to a clinician because ultimately people come in with symptoms and you want to treat them. If you have an objective measure that's non-invasive, but it doesn't vary as symptoms vary, then how useful will it be? And so basically what we show is that when we use our multi-electrode array and we extract spatial information, we can take a look at subjects and look at the percentage of time that the slow waves are not propagating towards the small intestines. That's on the x-axis. And on the y-axis is a clinically indicated symptom score of the severity of symptoms. So the larger this number, the more severe the symptoms. The stars correspond to controls, and what you can see is that basically everything is in the bottom left. So very seldom do you have abnormal slow waves, and also very seldom do you have bad symptoms. Patients who have gastroparesis, and notice that gastroparesis, sometimes they don't have many symptoms. Other times they have many symptoms. That shows that gastric emptying does not correlate with symptoms. That's been shown many, many times. Yeah, we can find the gastroparesis patients who have low symptoms, and we find their slow waves are not that abnormal, whereas other gastroparesis patients have very abnormal slow waves, and their symptoms are bad. So this is a measure that correlates with the severity of symptoms. So this starts to get exciting. And so some of the things that my PhD students are now beginning to work on is looking at, well, could we actually pinpoint the location of a spatial abnormality? Suppose we see one. And the reason that's important is because some of the treatments that they can do, for example, is people are building new types of pacing technologies and whatnot. And so one question we could ask ourselves is where should we place the pacing device? And so if I sort of separate the stomach, let's say for now, into 4 high-level regions, can I pinpoint the location of a spatial abnormality? And so the first question we asked was, well, suppose we have a CT scan on a subject. Then this is very much like what's done in neuroscience, where you can do source localization. If you have an anatomical MRI of the brain with EEG, well, what if we have an anatomical, location of the stomach based upon a CT scan? Can we actually solve the inverse problem? And so we attempted to do that. And so, for example, with 100 channel arrays, we did that. And one thing that's interesting about this for people who played around with source localization is this is definitely an ill-posed problem. There are many explanations that are consistent with the electrical recordings that you have. And so you have to impose some type of regularizer to enforce some solutions more so than others. And so that really boils down to doing Bayesian inference using an appropriate prior. And so we basically used a prior that has the property that it encourages solutions for which there's a small number of active bands, which we know occur, and further, it promotes them to evolve continuously over time. So it's almost like a Kalman filter combined with an L1 regularizer from a lasso. And so this prior, as I'm showing you here, has both of these properties. We published this in a Transactions on Signal Processing paper, but more importantly, we then used it in this PLOS1 paper. And what's nice is we can build very computationally efficient algorithms to find the Bayes optimal estimate. So when we do that, here's a plot of what we get. And so what you see here is every individual dot basically corresponds to the cutaneous surface. The color of the dot is the voltage. And then what we're overlaying or underneath there is the stomach, and you can actually see the activity, and you can see some of this propagation occurring. And so one of the things that we're actively doing is using these inverse techniques to help pinpoint the location of slow wave abnormalities. Now, another question that we got interested in is, well, a CT scan, maybe that was from a year ago or 6 months ago. And what if your stomach has moved since then? Can we develop a way to at least simplify the problem where we don't have imaging, but maybe we don't want to find the exact location of the slow wave abnormality. We just want to determine if there's a slow wave abnormality, turn it into a classification problem. And so my graduate student, Anjali Agrusa, basically has done this for part of her PhD. And basically what we've shown is that we can use deep learning ideas where we take the voltage at every electrode and we turn it into a color. And so since we have an array, that becomes a picture, and this is evolving over time. So this picture becomes a video. And then we use modern video classification techniques with convolutional neural networks. And we showcase that even if we don't place the electrode over the abdomen, and if we shift a lot in the vertical and the horizontal direction across many different BMIs, we can get robust classification performance. So that speaks to the sort of the key limitations, or the second key limitation. And the third one is the issue of dynamics. And, as people talk about all the time, sometimes when you show up to the doctor's office, you might have waited 6 months to get there, you don't present with symptoms. So when they assess you, it doesn't relate to what you feel a lot of the times. So we got interested in the idea of building an ambulatory device so you could actually take this device home with you and chronicle your symptoms. And so the high level idea is we know that the Brain Initiative got popular in the mid 2000s and late 2000s. And so there's all this emphasis on neurotechnology development. And so we basically just rode our surfboard over that wave of innovation. And it turns out that these signals that we care about are at the microvolt level, so very similar to EEG, although they're extremely low frequency, 0.05 hertz. So we can just slightly change some of these technologies for the Brain Initiative to make sure they're quote unquote DC coupled to capture low frequencies, and then implement our signal processing routines. And by doing that, we can do the artifact rejection, build a smartphone app to time synchronize when a bowel movement, a meal, a symptom occurs, and to look at all this information. And so to give a high level idea of what we can do is we can take a look at the power at the 0.05 hertz or the normalized EGG power and look at how it evolves within the context, for example, of a meal. And so we should, after a meal, we should see it increase. What are the details of how it increases? So we can focus on the time point if someone had a meal. We have a detailed description of what they ate, in this case, grape nuts and almond milk. We can see the time since they last reported a meal to make sure their stomach, should have been empty when they eat. We can look at all this information. We can build what's called the area under the curve, or basically throughout this duration of time, how much EGG power was there. So this is sort of amplitude and spectral information, but also, as I alluded to, spatial information is very important. And so one of the things that we also looked at in this example here is this red vertical line corresponds to when a symptom occurred. And notice that when a symptom occurred, the arrows in some sense are pointing to the right, which means retrograde or towards the esophagus. Whereas if you look at the two later blue vertical lines, they're in some sense pointing to the left, which is anterograde or towards the small intestines. So that tells us that this symptom right here is indeed associated with a gastric neuromuscular abnormality. And my colleague, Dr. Kunkel, emphasizes this is crucially important because it's unclear if these symptoms are actually due to a fundamental problem with the stomach, hypersensitivity. It could be the brain, it could be the colon. So if we can look at the time of a symptom and take a look at the electrophysiology, he can optimize his treatment regimen. And sort of as an example of this, to make this more clear, we actually were able to solve a GI motility case with this technology. And the high-level story is there's a 68-year-old man with bloating and early satiety, retired aerospace worker, played a lot of golf. His bloating was better in the morning, got worse throughout the day. His wife said at night he looks pregnant, and an overall feeling of being full and sick. Now, what makes this case interesting is that he also is a type 2 diabetic, and he takes a variety of medications to treat that. And the reason that's important is because we know diabetes can create neuropathies in your enteric nervous system or your vagus. And so when we go in and do the endoscope, my colleague couldn't even get the endoscope sufficiently down the stomach because it was filled with so much fluid and debris. When they sent him over to do that radioactive egg sandwich study, at the 4-hour mark, your stomach should be empty. 80% of the tracer was still in his stomach. So he's extremely delayed. When you do a CT scan on him, you can see his stomach looks bloated, just like a balloon. And so my colleague sort of brings forth this interesting thought process. Well, what would you do? You could give him erythromycin, which is basically a prokinetic, and that basically is just the gas pedal to the pacemaker cells in your antrum. You could try to get him something to better treat his diabetes. And as I mentioned, diabetes can create neuropathies of the vagus or of the enteric nervous system. You could try to, give him a breath test for small intestinal bacterial overgrowth. So that's looking at the duodenum, or go ahead and push to give him a diet for IBS, which is also related to the small intestines, or basically just try to treat his symptoms. Don't even try to treat the stomach. And so my colleague was strongly leaning towards his diabetes being the cause, because about half of diabetics have a gastroparesis. And so maybe his glucose maintenance was not the best. Or you could use this new ambulatory system. And when we use our new ambulatory system, you can see very rapidly that there's a huge, basically the pacemaker cells are always on. They're just extremely on. So he had a blockage. And so you can go in with pathology, and they can see exactly indeed that he had this very weird infection that you typically see in cows. And with that, the good news, though, is that it creates this sort of scar tissue in the pylorus area, the gatekeeper, and you can go in and do a very simple surgery. And so here's a CT scans after the surgery. His symptoms resolved. The EGG power sort of went back down. Everything looked nice. So another direction that we've been going towards is taking this ambulatory system that was kind of cute, but can we take it to the next level and use our stretchable electronics that were talked about early on? And so thankfully, the whole issue of locating the stomach can be resolved now with these little $2,000 portable ultrasound systems that can be plugged into an iPhone. You can locate the antrum of the stomach, place the array over the stomach, and we basically have shown that, before and after a meal, we see this increase in the area under the curve. In all subjects, we see a dominant frequency right at 0.05 hertz, everything as you would expect. And so this finding is under review for publication right now. So some ongoing work that we're pushing forward is in a couple of domains. One is if we focus on newborn babies, we call this project mouth to diaper. And at a high level is that we know that babies drink breast milk. There are novel technologies where you can quantify digitally exactly the input to the body. We can get the output of the body very easily. Babies pee and poop quite a bit. We can analyze the microbiome, and now we can place our device over the stomach. And we know that there are all these interesting feedback loops with short chain fatty acids and whatnot that modulate GI function. Can we track and watch how this occurs across human development? So we built some of our stretchable electrode arrays and tailored them towards the types of materials that go onto the baby skin. And we have a project with Lance Prince, who interestingly is the new division chief of neonatology at Stanford starting November 1. So we'll be continuing this project at Stanford, but we already have some preliminary results. We've been able to look at before and after a meal and see some interesting EGG activity. And our idea is to watch these dynamics across development. Now, moving forward to teenagers, we have an interesting project looking at eating disorders. And eating disorders is an interesting application because there's very much of a brain and a gut sort of connection. And so I collaborate with these two distinguished faculty, as you see here, who both are experts within this topic. And one of the things that people may or may not know is that anorexia has the highest rate of death of any psychiatric illness. And so at a high level, what's interesting about the brain-gut axis is that we know that anorexia is this low weight, fear of weight gain, distorted body image, and this can give rise to deficits in interoception of your viscera, or the information coming from your stomach as well as your heart. And there's also a new DSM-5 categorization for ARFID, which basically is this visceral hypersensitivity. So as you take in food, you feel lots of pain, and as a consequence, you avoid eating lots of food, and this creates all sorts of craziness in gut motility. And so during weight restoration, there are all these GI complaints that arise, and motility is very jacked up. And so one of the things our technology can do is it can measure the dynamics of gastric neuromuscular function during this recovery, and we can relate these dynamics to clinical psychological changes, and it can create all sorts of interesting interventions with biofeedback, for example, to increase interoception. And so we have some preliminary data on this that's interesting, where, and what I'll just focus on is where this last blue vertical line is here associated with the water load test. One thing I forgot to say about our electrodes is that we get the peaks of the EKG for free. If you place electrodes on the abdomen, you see our peaks of the EKG for free. So we can get heart rate information as well as the gastric information. And we see these interesting dynamics in both right around when the water load test occurs. And the water load test is a way to test your interoception, because you're supposed to drink water until you're full. And how well can you have this sense of fullness? That's a measure of interoception. But also, as I mentioned before, when those stretch receptors occur in your stomach, there's these vagal afferents that get passed to your body. But also, when your stomach gets more things that come into it, there's a sympathetic input to basically increase blood flow to your mesenteric artery. So there are all these interesting dynamics that occur, and this led to an interesting project that we wanted to do, where we're going to be doing brain gut monitoring next. But as an intermediate step, what we wanted to do was to look at the heart rate variability, or the sympathovagal ratio, the ratio of sympathetic and parasympathetic, and look at how it aligns temporally with the EGG power. And one of the things that we find is that at every local minimum of the sympathovagal ratio, so where there's basically a local maximum in parasympathetic activity, we see lots of EGG activity, as you would expect. So we can look at the exact time points of when these local minima in the parasympathetic, or the sympathovagal ratio occurs. So that's a local maximum in parasympathetic, and look at the time points of how this relates to EGG power. We can turn these basically into binary signals, and then we can implement some new algorithms that my research group has done with causal inference to see what is the information flow. Is it going from the brain to the gut, or from the gut back to the brain? And basically what we find is that in this example, if you look at these axes, the first plot is across time, the gut going to the autonomic nervous system versus in the other direction. And we see there's much more information flow from the autonomic to the gut as we anticipated. And so this just provides a sense of how these problems bring forth new interesting research questions. And so for the purpose of fast through Linda Nguyen at Stanford, who's coming up with novel treatment regimens, and she has a clinical trial on using vagus nerve stimulation to increase parasympathetic activity, which remember increases the gut. And we have evidence that we can see that when we use simultaneously our EGG devices on some of her patients. So as you can see, there's a huge opportunity for population health opportunities. We now are in the process of doing simultaneous EEG and EGG monitoring, and the brain and the gut are connected in so many interesting ways. And so we think there's huge opportunities with the advent of smartphone apps to digitize what types of food you're intaking from just taking a picture. We have a digital description of the function of the stomach. Combining this with brain measures, we can start to look at the interplay between all of these different things. And so As I conclude my talk, I like to just give this quote that sort of speaks to the philosophy of how we like to think, and that is, why do we call them red oceans? And so there's red oceans and blue oceans. Red oceans are where there's cutthroat competition in existing industries, and it turns the ocean bloody red. And we look at sort of this project in this GI domain as a blue ocean because it's for much less explored and thus untainted by competition. And so it allows people in our research group to move slowly and carefully and not have to worry about clicking the refresh button on Google Scholar to make sure they didn't get scooped. And yet it has very important and profound implications to improve human health. So with that, I conclude my talk. Thank you.
Todd, thank you so much. That was fantastic. We have a whole stream of questions for you, and let me just curate those a little bit. From Krishna, what might be the opportunities to use your beautiful new techniques in combination with swallowable e-pill based sensors, simultaneously intra-GI tract chemistry and imaging together with non-invasive electrophysiology?
Yes, I think there are many interesting opportunities. Thanks for that question, Krishna. And so there already exists some pill technology. So, for example, there's something called the smart pill that is used sometimes in GI. I think if you combine that with the electrophysiology, there's many things that you could do. One issue with the smart pill that many GI docs have is that it provides information about what's occurring over time, but you don't know the exact location of the device. You can envision, and again, this is me wearing my electrical engineering hat, you could use a multi-electrode array for the purpose of doing localization to pinpoint the exact location. Now you know exactly where it is as you're measuring all these readouts. There's all sorts of exciting things that you can do in looking at the chemistry that's occurring. Also, you can look at the contractions or the mechanical information that's taking place. And there are even some people that are now actually being able to try to sample the microbiome as it propagates through your digestive system. As people may or may not know, when you do the stool sample sort of technique, the problem is that when you look at that stool sample, you don't know what part of the digestive system those microbes are. And it turns out that the composition of microbes in our digestive system is very spatially dependent. And so it varies a lot spatially. So combining all these things together could definitely be fascinating. Yes.
Yeah, cool. A question from Bill Newsom. Is it possible to extend the measurements below the stomach into the duodenum?
Yes, and so we have some preliminary results on the duodenum, and the duodenum is very exciting because you get to talk about the pancreatic ducts. You get to talk about when you're looking at issues of glucose fluctuations and diabetes. All of that is in the vicinity of the duodenum, and so it turns out the duodenum is not that far from the stomach, and it's if you take a look at where the electrodes are, it's quite close. Thankfully, the pacemaker cells in the duodenum are at about 0.2 hertz, whereas our cells in the stomach are 0.05 hertz. So it's kind of very much like in the ear where we say God did a Fourier transform. In some sense, God is doing a Fourier transform with the pacemaker cells of our digestive system. And so we actually have some preliminary results showcasing that we can do it. But that's an active area of exploration. Yeah.
Cool. Another question from Carla. Hey, Todd. People with spinal cord injury have severe GI problems. Do you think your approaches would be helpful? And do you have any plans to examine them?
I've heard that before, and absolutely I would. And so, for example, one big issue is the urge to void and whatnot. And if you could have any information ahead of time suggesting that there's a need to void, could you help as a tool in doing this? And so I'm only constrained by the amount of time that I have, but I love great ideas. And so if I find the right partnership, I would love to explore it. Yeah.
Yeah, thanks. A question from a famous researcher named Anonymous. What is the balance of information you get from the details of the time series signal versus the summary statistics like area under the curve?
Excellent question. So I reported on different things. And so the area under the curve is primarily looking at the power in a specific frequency band, sort of across time. So that in some sense is telling us about if those pacemaker cells, are they at least, is there membrane potential going up and down? Is that neurotransmitter like the acetylcholine coming from enteric neurons after you eat a meal? Is that operating correctly? It's telling us those things. But it could be that that information is correct, but what if the spatial pattern, the orientation of these guys is messed up? That can occur. And so what we now do is we're building almost like a report that has summary information across multiple aspects, one of which being that information, as I just alluded to, about the area under the curve, the other about the percentage of time the slow waves are operating in a wrong direction. And my graduate student, Anjali Agrusa, who I'm giving shouts out to, she has some very exciting new work on building the appropriate summary statistics of the spatial information. And these are circular statistics, right? So 0 and 360 are the right thing. So there's all this fun applied math that we can do to come up with, in some sense, the right notion of an average. And so it's a notion of a barycenter, et cetera. And she has new findings that showcase that when we use these new techniques, they separate sort of very well. And so that's an ongoing project. Yeah.
Cool. We make lots of measurements of EMG from leg muscles, and it's challenging for us to deal with subcutaneous fat. I'm wondering if how the fat affects the signals you detect on the surface of the abdomen.
Excellent question. And so it turns out in our publication, our first clinical journal paper that appeared in December 2019, where we showcased that the slow wave, the spatial slow wave abnormalities spatially correlate with the severity of symptoms. We had patients with BMI up to 36, and we were able to robustly extract that information. And so it looks like being able to extract the spatial slow wave information, because we know exactly it's at this very narrow band right around 0.05 hertz. It's quite robust to BMI. Yeah.
Okay, great. That's helpful to know. Here's kind of a combined set of questions, Todd. The first one you began to answer, and the second one's new. So how can you combine your information from the wearable sensors with other information like clinical record, heart rate, labs, to create a more robust statistical model? And the second question related, how do you know when your model is sufficiently accurate?
Okay, great question. So the first one is, how can we combine other pieces of information? Absolutely. And so, so for example, one of the things that Lyndon Nguyen does, who I alluded to, who leads the GI motility project at Stanford, she actually, not all GI docs do this, but she actually does big bouts of autonomic testing on her patients, because sometimes what you find is that if the brakes and the gas pedals to the digestive system are not operating accordingly, it doesn't matter if the engine is working perfectly. And so should the focus be more on the brakes and the gas pedals, or should it be on repairing the engine? And so without question, we like to get, for example, as I mentioned, that sympathovagal heart rate information, which is a proxy. It's not perfect, but it's a proxy for the sympathetic parasympathetic nervous system. We like to get that and combine that with this information. So if we can start to see trends that suggest what's causing these symptoms is A versus B versus C. And so another great thing that you can get as an example is if you can get blood work in people, then you can actually, so we know that inflammation is tied to our sympathetic nervous system very well. So what you find in many patients who have what's called functional dyspepsia, which is they have all these symptoms, but their gastric emptying is normal. And a lot of situations, what you find is that there's all sorts of inflammatory markers you can see in the blood. And that's suggestive that if you have an overemphasis of these inflammatory markers, you're going to have increased sympathetic activity that's just hitting the brake pedal on your digestive system. And then you can come up with ways to try to address that. So it's a very complicated problem, but that's also what makes it fun as a data scientist. Yeah.
Yeah. So Todd, just one last question from a fellow technology developer. Is the array big enough? Do you need a higher density array, a larger array? It looked like you were missing the stomach on one. Where are you with that? And do you need more? Would that give you better input to your models?
Excellent question. So I have, and I think this relates to another question I didn't ask, but one of my PhD students, Alexis Allegra, she's a PhD student in electrical engineering. The focus of her thesis is on doing a detailed sort of theoretical analysis using ideas from estimation theory to determine what should the optimal array density be. And there's all sorts of trade-offs, because if you make the electrode smaller in area, then you can cover with a higher density array. But then the impedance of the electrode is basically inversely proportional to the array. So in some sense, you have a bit larger noise variance. So there's all these interesting trade-offs, and that's actually part of her thesis. In terms of the array not being over the stomach, those 3 red dots were not over the stomach, but those 3 red dots came from the 1980s perspective. moving forward on all of our subjects, we always do imaging to make sure that we're covering the
I
antrum.
see. Yeah. Yeah, it's great having that cheap ultrasound too.
Yeah, thanks to the people who developed that. Yeah,
Yeah.
Butterfly Networks. Yeah.
Yep, really nice. Todd, I just want to thank you again. It's super interesting. I love hearing your lectures. I always get a bunch of new ideas. I actually sent you a few emails with some ideas while you were talking, and I'm just really pleased you could be here today, and thrilled that you'll be joining us at Stanford as a colleague in bioengineering in the Wuxi Neurosciences Institute. So thank you very much. Appreciate it.
Thank you. Thank you.